Good morning. Let us check a few important 2 marks and 3 marks questions. But before that, my good wishes for your board exams. Please note, do not forget to check the various diagrams, different flow charts, different images from your Ncert textbook. A lot of diagram-based questions are coming. So try to get hold of those. Okay. So let us begin. The first question, it says, identify the chemical structure of the compound shown here. State any 3 of its physical properties. So the compound which you see here is that of morphine. Okay. And you can write it's white, odorless, and a crystalline compound. Additionally, many questions do come from some other facts, such as what is the source of morphine? So you can write morphine is extracted from the latex of poppy plant, that is Papaver somniferum. Also, they ask about heroin. So what is heroin? Heroin is basically diacetyl morphine, and how it is prepared? It is basically prepared by acetylation of morphine. So you have a natural compound and you acetylate it and you get heroin. It is also known as smack. How is heroin taken? Heroin is basically taken by snorting and injection. Which class of substance does it belong to? It's a depressant. Please note that, right? Then, humans have innate immunity for protection against pathogens that may enter the gut along with food. What are the 2 barriers that protect the body from such pathogens? The question says that when we eat food, some pathogens can also enter the gut, right? But we have the innate immunity for protection against those pathogens. So we have to cite 2 barriers. So the 2 protective barriers are the physical barrier and the physiological barrier. So on the physical barrier, you can mention about mucus coating of the epithelium that lines the gastrointestinal tract. What does this mucus do? The mucus basically helps in trapping the microbes that enters the body, right? And when you talk about the physiological barrier, you can mention about the acid in the stomach and saliva in the mouth. This prevent the microbial growth, okay? Then a question which integrates a lot of different concepts like sewage treatment, then pollution, biochemical oxygen demand, etc., etc. So let us check the question. Water samples were collected at points A, B, and C in a segment of a river near a sugar factory and tested for BOD level. Fine. The first statement says that at 3 points, A, B, and C, different water samples were collected, right? Where? In a segment of a river. And this river was near a sugar factory, okay? And the various samples, that is the water samples, were subjected to BOD assessment. We'll check what is BOD, but let us continue with that question. According to the question, it says the BOD levels of the samples A, B, and C were 400 mg per liter, 480 mg per liter, and 8 mg per liter respectively. What is this indicative of? So you see that sample A and B recorded very high BOD, while sample C, the BOD level was quite low. Then we have to write what does this indicate, okay? The second part of the question says, explain why the BOD level gets reduced considerably at the collection point C. So we have to explain this observation. So before that, please have a look what is BOD. So you might be knowing that because of different industrial activities and different domestic discharge, the water bodies get polluted. Okay. So when we talk about organic pollutants, the microbes, particularly the bacteria, as well as other microbes, they need oxygen, right, for the degradation of the organic pollutants under a specified temperature and a given time period, so that the organic pollutants can be degraded and the water body becomes clearer and cleaner. So biochemical oxygen demand is the amount of oxygen used by bacteria and other microbes during the process of decomposition of organic matter under aerobic conditions. Okay, oxygen is required at a particular temperature and in specific time period. In other words, it basically symbolizes the amount of organic pollution present in an organic aquatic ecosystem. So higher the BOD level, greater is the organic pollution level. So in simple words, that's what it is. So according to the question, you see the diagram. First, you have a river. There are different points A, B, C, etc. So there's a sugar factory at the upstream. Also, near point B, there's a sewage discharge point. So both uh, the sugar factory and the sewage discharge point are basically responsible for polluting. Okay, they are discharging a lot of organic waste into the river. Obviously, the BOD level will be high. So that's what we have. For the first part of the question, you can write at collection points A and B, the BOD level is high due to high organic pollution caused by the sugar factory and the sewage discharge. Okay. Now, when you talk about point C, so if you carefully look into the diagram, you have a sewage treatment plant, okay, STP. So in sewage treatment plant, you subject the water to different levels of treatment, primary treatment, secondary treatment. So when you talk about secondary treatment, a lot of different useful aerobic microbes are involved. They consume the major part of the organic matter present in the river water or effluent. Fine. So as a result, the organic pollutant level goes down and the amount of organic pollution is lesser. So the BOD level also goes down. Next question. A patient admitted in ICU was diagnosed to have suffered from myocardial infarction. Condition of coronary artery is depicted in the image below. Name two bioactive agents and a mode of action that can improve this condition. Here we have something called myocardial infarction. In other words, it refers to heart attack. So basically, you know that heart attack is a medical emergency. And when does it occur? It usually occurs when blood clot blocks blood flow to the heart. And as the heart muscle is not receiving the blood flow, the tissue loses oxygen and it dies. That's what a myocardial infarction is all about, right? Now, if you carefully check the diagram, you can see there are cholesterol plaques and there's a blood clot. Obviously, the blood flow is obstructed, right? So in order to assist or help the patient, the following two bioactive compounds or bioactive agents can be administered. Number one, streptokinase. So please remember, streptokinase is an enzyme. It is produced by the bacterium Streptococcus, and it is often popularly called as clot buster because it assists in removing clots from the blood vessels of patients with myocardial infarction. Okay, then the second bioactive compound are the statins. They are basically blood cholesterol lowering agents, and they're produced by the yeast called Monascus purpureus. So you have streptokinase and statins. If administered to this patient, they might help him or her. Then substantiate by giving two reasons as to why a holistic understanding of the flora and fauna of a crop plant is required before introducing an appropriate biocontrol method. So basically what this question asks, you might be well aware of the fact that a lot of different pathogens, different pests, different diseases cause huge loss to the agricultural crops. And we often thrive upon different pesticides, different insecticides to protect our crops. However, there are a lot of price that we have to pay when we use this insecticide, pesticides, etc. So basically, this insecticides and pesticides, they are harmful to the environment. They can enter the food chain. They can into our system, and that can lead to a lot of havoc, a lot of diseases, a lot of unwanted consequences. Secondly, they're costly. Okay, so the farmer has to pay a lot of extra money. Now, what if we can use natural agents or say some modified organisms to ward off the pathogens and diseases? So basically, we're trying to use biological control agents, which are say natural enemies of the pathogens, and we are able to protect the plant, right? So that is the essence of biocontrol agents or biological control agents. So they can be basically defined as the use of natural efficient strains of any organism or organisms that reduce the incidence of severity of diseases caused by plant pathogens. It exhibits an antagonistic activity towards a particular pathogen. Now, the thing is that if you introduce a biocontrol agent without actually knowing what kind of intricate or complex food chain is there in the cropland, then it might usher in unwanted effects. Okay, see, there are a lot of beneficial predator-prey relationship that happens or that are naturally operative. So if we are interested in eradicating the pest in a crop field, then this might also disrupt the predator-prey relationship, right? So many beneficial predatory and parasitic insects, okay, which depend upon the flora and fauna of the crop field as for food or host, they may not be able to survive. And this is not what we want, right? Because that will wipe away the beneficial insects. For example, honeybees might be eradicated if we introduce some enemies of honeybees, okay, or some beneficial microbes which say are involved in nourishing the soil. And if we introduce some other microbes that might release some chemical agents that will kill the beneficial microbes, that is also not what we expect, right? So if we have a proper understanding or holistic understanding, so holistic approach basically is an all-encompassing approach. Means you try to understand each and every aspect. So it ensures that various life forms that inhabit the field, their life cycles, patterns of feeding, the habitats that they prefer are extensively studied and considered. So that's why a holistic understanding of the flora and fauna of a cropland is required before introducing an appropriate biocontrol method. So which biocontrol method will be appropriate? In order to introduce that approach, we should know the flora and fauna of the cropland. Then an ecologist is studying an area. There's a population called A, and this population A is surviving on unlimited resources. So it shows exponential growth. Now, populations B and C are introduced to the same area. What will be the effect on the availability of resources, interaction, and growth pattern of the populations A, B, and C when they start living together in the same habitat? So basically what the question says, there's an ecologist, he's studying a particular area, and that area is dwelled in by a population called A. Okay, and it also says the population A is surviving on unlimited resources. So if you check the diagram first, that's given on this right panel, you have, the population density on the y-axis, and on the x-axis you have the time. So the red colored, or rather for you might appear pinkish red, you have a J-shaped plot, basically shows an increase in population when the resource available is unlimited. Population A is having this pattern. So this is basically an exponential growth pattern. Now, when we talk about the introduction of populations B and C, what will happen? There will be a lot of competition among the individuals of population A, B, and C. So the limit of resources will be reached. I mean, the resources won't be unlimited. So the survival of the fittest, that concept will be now operational. So what happens in the first phase? The interaction among the populations A, B, and C will lead to the competition between the individuals. And eventually the fittest individuals will survive and reproduce. Now, as the resources will become definitely finite and limiting, the population growth will become more realistic. So there, on final stabilization, it will be somewhat like a J-shaped plot turning into an S-shaped plot, that is this blue graph. So you have this lag phase, then there will be a growth phase, and finally it will no longer increase. It will reach a limit, and that is the carrying capacity. This is the logistic growth, and it is more realistic. When resources are limited, the populations generally show logistic growth pattern. Next, with the decline in the population of fixed species, it is noticed that the population of wasp species also starts to decline. What is the relationship between the two, and what could be the possible reason for decline of wasps? So basically it says that there's a fig species, and if the population of the fig species is on decline, then the population of wasp also starts to decline. So this is indicative of a mutualistic kind of relationship, and fig depends on wasp for pollination, and wasp depends on fig for food and shelter, right? So simple, the question itself is elaborative. With the decline in population of figs, basically the wasp loses its source of food and shelter. That's why there is a one-to-one correspondence between the decline of the population of either species. Next, with the increase in the global temperature, the individuals of different species in Antarctica, they are facing fluctuations in the temperature. Okay. Out of the regulators and conformers, which of the two will have better chances of survival? Give two adaptations that support them to survive in the ambient environment, and give one suitable example. So here we see that the question mentions about fluctuations in the temperature in Antarctica. And you also might be aware of the fact that an individual interacts with its environment, both biotic and abiotic factors. And the environmental factors, they change. For example, the temperature, okay, the osmotic pressure, the osmotic concentration, right? So the organisms always try to maintain or maintain a homeostatic status. Okay. So when they try, some of the organisms, they are able to maintain an internal environment despite changes in the outside environment. Okay. So those type of organisms are known as regulators. Okay. And some of other organisms, they are unable to maintain a constant internal environment and change according to the ambient conditions. Okay. These regulators, they show a much wider range of distribution, while conformers, they show a narrow range of distribution. Okay. So this concept should be clear: regulators and conformers. So once again, I repeat, regulators are basically able to regulate or maintain a constant internal environment despite the changes in the external environment, while conformers, they fail to maintain a constant internal environment and change according to the ambient conditions. Okay. In the diagram, you can also see about partial regulators. That is, to an extent, they try to change their internal environment according to the atmospheric conditions. But if the changes in the external environment keep on increasing, then they are able to stabilize or maintain a constant internal environment. Okay, so those are partial regulators. So here it says, out of the regulators and the conformers, which of the two will have better chances of survival? So the regulators, we know they are able to withstand fluctuations. So obviously regulators will be in a state of better survival. The second part asks, give to adaptations that support them to survive in the ambient environment. So they can either regulate the temperature, that is thermoregulation, or they can opt for osmotic regulation or osmoregulation. Then give one suitable example. Birds and mammals are regulators, while you also might be asked, give examples of conformers, reptiles, amphibians, and all those stuff. Then, how do normal cells get transformed into cancerous neoplastic cells? Elaborate, giving 3 examples of inducing agent. So cancer, in simple words, uncontrolled cell division, right? And you can also imagine the whole concept of cancerous cell as a kind of terrorist in the cellular society. Okay, simply put like that. Now here there's a small word called neoplastic cells. So when you talk about neoplasia, neoplasia is basically the new uncontrolled growth of cells that is not under physiologic control. So the whole physiological control is out of gear when neoplasia sets in. So the transformation of normal cells into cancerous neoplastic cells can be induced by various physical, chemical, or biological agents. These agents can cause DNA damage. There are some genes called cellular oncogenes or proto-oncogenes. When active under certain conditions, it results in cancer. There are different sorts of inducing agents that can actually lead to the transformation of normal cells into cancerous neoplastic cells. Some of these are cited here. You can mention about ionizing radiations like X-rays and gamma rays. Then you have non-ionizing radiations like UV. Then there are a lot of chemical carcinogens, example those present in tobacco smoke. Also, please note there are several viruses that can lead to cancer. For example, the human papilloma virus can cause cervical and several other cancers, and also hepatitis C can lead to liver cancer and non-Hodgkin's lymphoma. That's it. Next, a person is suffering from a high-grade fever. which symptoms will help to identify if he or she is suffering from typhoid, pneumonia, or malaria. First, sustained high fever, weakness, stomach pain, constipation, headache, and loss of appetite are suggestive of typhoid. Fever, chills, cough, and headache, lips and fingernails turning gray to bluish are suggestive of pneumonia. If the person experiences chills and high fever recurring every 3 to 4 days, then he or she might be suffering from malaria. However, please note, tests are required. Okay, otherwise, wrong diagnosis without testing may lead to disaster again. So, proper medical intervention is necessary. And also, I want to mention here, please check the causative organism for these diseases. And do not forget to about the mechanism, how the malarial parasite functions. Then, recognition of an antigenic protein of a pathogen or exposure to a pathogen occurs during many times of immune responses, including active immunity and induced active immunity. Specify the types of responses elicited when human beings get encountered by a pathogen, right? This is a 3-mark question. So basically, you can check this diagram on the right-hand side. So you have on the y-axis the antibody concentration and the x-axis the number of days or time. Okay. So suppose at this point you have the exposure to an antigenic protein, say A. Okay. So whenever there is an exposure to the protein A, imagine that an advertisement goes through your body that we are under attack. Okay. So the body will try to respond. This is the first initial exposure, and the body will exhibit the primary immune response. During this phase, there are a lot of naive B cells on exposure. They will be activated, and initially they will be converted into short-lived plasma cells, which will release some of antibodies, and these antibodies will try to ward off the antigenic protein A. Please remember the antibodies during this phase are mostly IgM, but also some IgG antibodies are produced. Okay, so this is the primary immune response. Okay, so here we have mentioned when our body encounters an antigenic protein or pathogen for the first time, it produces a response. The important facts are the first primary immune response is of low intensity, but it retains a memory of the first encounter. Okay, so whenever this initial exposure to A happens, there are few antibodies which will be responsible for remembering them. Okay, they remember the enemy. Okay, so when there is a subsequent exposure, suppose this is say on the 25th day or 26th day, again the pathogen has attacked you, then this time what happens, the immune response is quite intensified. Okay, and different special types of lymphocytes present in our blood called B lymphocytes and T lymphocytes come into play. And we also had mentioned that there is a memory part of the whole immune system. So this memory B cells will differentiate into highly active plasma cells, and these plasma cells will release a lot of antibodies. Okay, so this the B lymphocytes, when they release these antibodies, they will act as the army. Okay, so this whole aspect is the secondary immune response, and also it is seen that IgGs are the main antibodies secreted with some small amounts of IgM sometimes. Okay, so this are the different steps that are often observed during the primary immune response and the secondary immune response. That's it. Next, in a pathological lab, a series of steps were undertaken for finding the gene of interest. Describe the steps or make a flow chart showing the process of amplification of this gene of interest. Okay. So here there are a few words like amplification of gene. Okay. So in simple words, we can state that gene is a segment of DNA which gets transcribed and translated. Okay. So here in a pathological lab, different steps were undertaken to find the gene of interest. So if we want to amplify it, okay, then what technique can we use? Answer is PCR, polymer chain reaction. And what are the different steps? So first, let us check the diagram. Suppose the double-stranded DNA is shown in the first step, and the red colored boxes represent the nucleotide sequence in the gene of interest, and we want that it has to be amplified. So first we need to denature it. How do we denature? That is, the DNA strands have to unwind. How? By subjecting the strands to heat treatment at around 94 degrees Celsius. And this results in the separation of the strands. That is the denaturation step. Then we have the second step called annealing. So here the primers anneal to the complementary strands, the RNA primers. You can see here this step. Then we use the DNA polymerase enzyme, particularly the Taq polymerase, with the nucleotides, that is the deoxynucleotides and divalent ions, so that the extension of the strands is facilitated. So these are the three steps: denaturation, annealing, and extension. So say after 30 cycles, we might have around 1 billion copies of the gene of interest. So this is polymerase chain reaction. Next question states, Evil Quartet describes the species extinction or loss of biodiversity due to human activities, right? Explain how the population of organisms is affected by the fragmentation of the habitat. Part A. Part B says, introduction of alien species has led to environmental damage and decline of indigenous species. Give any one example of how it has affected the indigenous species. Number 3, or C, could the extinction of stellar sea cow and passenger pigeon be saved by man? Give reasons to support your answer. Okay, so there's a concept of evil quartet. So it mentions about habitat loss and fragmentation, co-extinction, alien species invasion, and overexploitation as the four major backdrop or reasons why there's loss of biodiversity. Okay, so when a large habitat is broken into small fragments due to various activities, the mammals and birds requiring large territories and certain animals with migratory habitats are profoundly affected. So this leads to their population decline. Okay. Then the question part B mentions about the introduction of alien species and how they are affecting indigenous species. So some of the examples are cited here. Nile perch, it was introduced in Lake Victoria, and it resulted in the extinction of an ecologically unique assemblage of more than 200 species of cichlid fish. Okay. And Perthenium or carrot grass, lantana, water hyacinth. Okay. They spread across different territories very fast, and they cause a lot of environmental damage and is a threat to our native species. Okay, carrot grass, water hyacinth, lantana. These are very common. You can check all around. Then also you have something called African catfish. It was basically introduced for aquaculture, but it turned out that it poses a huge threat to the indigenous catfishes in our rivers. So any one example you can cite here. The third part says, could the extinction of Steller's sea cow and passengers pigeon be saved by man? Yes, humans have overexploited natural resources for the greed rather than need, leading to extinction of these species. Sustainable harvesting could have prevented extinction of these species. Okay, sustainable development, sustainable management, sustainable use, sustainable agriculture, sustainable harvesting, all these terms are very, very critical, very important, particularly in today's context. Then, the image below is of a sacred grove found in India. Explain how has human involvement helped in the preservation of these biodiversity-rich regions? The second part of the question says, value of Z or regression coefficient is considered for measuring the species richness of an area. If the value of Z is 0.7 for area A and 0.15 for area B, which area has higher species richness and a steeper slope? So the image which you see here represents a sacred grove. Okay. It's a way of conserving biodiversity. It's a way to protect nature. Okay. So if you happen to visit Northeast India, there's a state called Meghalaya, and sacred groups are great tourist attraction. Okay. So India's long history of religious and cultural traditions have focused on the protection of nature. Okay, different states. Okay, in many cultures, tracts of forest are set aside. All the trees and wildlife within are venerated, respected, and they are provided total protection. Sacred groves in many states are the last refuge for a large number of rare and threatened plants. For example, sacred groves in Meghalaya represent a long tradition of environmental conservation based on indigenous knowledge by the tribal communities. In these forests, cutting of trees, plucking of flowers, fruits, twigs are not allowed. They serve as a natural treasure house of biodiversity and a refuge for a large number of endemic, endangered, and rare taxa. Right? Now, when you talk about species richness of an area, let us consider this plot. So the y-axis, we represent the species richness, and on the x-axis, we have the area. Normally, the relationship between species richness and area will give you this rectangular hyperbola represented in blue. S is equal to C a to the power Z. Okay, where S represents species richness, A is the area, C is the y-intercept, and Z is the slope of the line or regression coefficient. Okay, so S is equal to C a to the power Z. If we want to write it in logarithmic scale, the relationship is a straight line, which you see here. That is this log scale. Log S is equal to log C plus Z log A. Y is equal to mx plus C, straight line equation. Now, considering this value of Z is very critical. Okay. Generally, for small areas, the Z value is around 1.2 to 0.1 to 0.2. But for large areas, example, the entire continents, the slope of the line is steeper. Z value ranges from 0.6 to 1.2. Example, for frugivorous, fruit-eating birds and mammals in the tropical forest of different continents, the Z value is 1.15. Okay. So here the question says the value of Z is 0.7 for area A and 0.15 for area B. Okay. So which area has higher species richness and steeper slope? So obviously the value of Z is higher for area A. That implies it is steeper, and correspondingly the species diversity, or rather the species richness, is higher. So area A will have more species richness and a steeper slope. That's it. The image below depicts the result of a gel electrophoresis. If the leather represents sequence length up to 3000 base pairs, which of the bands 1 to 4 correspond to 2500 base pair and 100 base pair respectively? And part B, explain the basis of this kind of separation and also mention the significance of this process. Okay, so to separate DNA using Agarose gel electrophoresis, the DNA is loaded into precast wells in the gel and a current is applied, right? So this is a gel, suppose, and these are the wells. Okay, here we load the different samples, and as a reference we have this leather. So you also might be knowing the fact that the phosphate backbone of the DNA molecule is negatively charged. Therefore, when placed in an electric field, the DNA fragments will migrate to the positively charged anode, right? Then, next important fact is the DNA fragments will separate, or to state in other words, resolve according to their size through the sieving effect provided by the agarose gel. That means the gel, the material is agarose, which is normally extracted from different seaweeds, uh, Gelidium, gracilaria, etc. And agar. And you have a sieving effect of the gel. Okay, because of the cross-link between the polymeric chains, there are certain porous structures within, and the DNA threads tend to move through these pores according to their size, okay, on application of electric field. However, the important fact is smaller the fragment size, the farther it moves from the point of loading. So if you are loading your sample in this well, if the size of the DNA fragments is smaller, it will move farther. If the size of the DNA sample is big or large, then it will move to a lesser distance. Okay. Now here, the leather represents sequence length up to 3000 base pairs. So these are small sequence and these are longer segments. Fine. So the length is 3000 base pairs. Which of the bands 1 to 4 correspond to 2000 base pair and 100 base pair respectively? So band 3 we see is here and it is nearer 3000. So this might correspond to 2500 base pairs, and band 4, which is the farthest, it is the smallest and it might be corresponding to 100 base pair. Okay. Why do you interpret it that way? Because the fragments will resolve according to their size. The smaller fragments will move further from the well. As seen, band 4 represents the smaller fragments as compared to band 3 corresponding to 2500 base pair. Now, why do you need this kind of separation? Okay, or what is the significance of electrophoresis? The significance of electrophoresis may be cited as number one, to check the quality and quantity of genomic DNA after DNA extraction. Means you have extracted the DNA, whether the quality and the quantity of the DNA is fine enough for further processing, for further steps. You can also mention about the use of gel electrophoresis to purify the DNA fragments for use in constructing recombinant DNA by joining them with cloning vectors. Fine. That's it. So in the next session, we'll come up with few more questions, longer questions. Till then, have a great study time. All the best.